In this lecture, we're going to look at viral pathogenesis from the point of view of dissemination, virulence, and epidemiology. Now, of course, pathogenesis is the entire process by which viruses cause disease. With virulence, we're talking about the capacity of a virus to cause the disease. And dissemination, of course, is the spread of the disease. There are fundamental questions of viral pathogenesis that we have to consider. So how does the virus enter the host? What's the initial host response? Where does primary replication occur? How does the infection spread in the host? What organs and tissues are infected? How is the infection transmitted to other hosts? And is the infection cleared from the host, or is it a persistent infection? So how these questions are answered determines what the pathogenesis of the virus is, and we'll see that different viruses show different patterns of pathogenesis. So one of the key factors in studying a disease is to come up with answers to these basic questions. So first, let's look at viral entry. The main ways that viruses enter a host is through the respiratory tract, the alimentary tract, the urogenital tract, the eyes, or the skin. So we'll look at these individually. The respiratory tract is probably the most common route of viral entry for viruses, even for viruses that we don't necessarily associate with being respiratory viruses. We have 140 square meters of absorptive area in our respiratory tract. It's so large because when you go down into the lungs and you get to the level of the alveoli, we have an incredible amount of surface, absorptive surface, for gas exchange. The resting ventilation rate for your average person is about 6 liters of air per minute. And we have 300 million alveoli. So that's a lot of material moving in and out. So it's easy to see why this would be such a common route of entry. Now, mechanical barriers are important. They're ciliated cells, and we also produce about 100 ml of mucus per day. Remember the mucociliary escalator that we've already talked about. Now, when you get down to the level of the alveoli, they don't have cilia or mucus, but there are macrophages lining them that can act to ingest particles. There's coughing and sneezing. That's important. That produces aerosols of particles from 1 to more than 20 microns in size. This permits infection at a distance. Dispersion of an aerosol depends on wind currents and particle size. A particle of about 100 micron diameter requires 10 seconds to fall from the height of the average room. 20 micron particles require 4 minutes. 10 micron particles require 17 minutes. So the smaller the particle, the longer it can remain in the air, suspended in the air after a cough or a sneeze or a shout or any other kind of mechanism that can result in the release of particles from the respiratory system. Now, mechanical barriers are important. Larger particles get trapped in the nose. When you think about the hairs in your nose, they're not just there for decoration. They're actually acting as strainers. So you have mucus in the hairs in your nose providing a good physical barrier to materials coming into your respiratory system. And that's part of the reason why they recommend, for instance, that you try to avoid breathing through your mouth too much, because the nose provides such an important barrier to all kinds of microbes and dirt and dust and so on that you could be inspiring. Now, smaller particles will not be trapped, and they can make it deeper into the respiratory system. But just to give you a feel for how effective this can be, one study found that nearly 2 million virus particles could be released in a single sneeze, and 90,000 from a cough. I mean, this is what we're dealing with coronavirus, why people are wearing masks, because we are so efficient at releasing virus particles in from our respiratory tract through sneezing, coughing, breathing, talking, you name it. Now, we do see that some viruses will be localized to the upper respiratory tract and some to the lower respiratory tract. The upper respiratory tract is where we typically will find rhinoviruses. They get their name rhino from rhino for the nose. And these viruses cause probably about half of the colds that we get every year. And that's part of the reason why colds tend to be not so serious, just more annoying than anything else, because they tend to be localized to our upper respiratory tract. Now, the flu, which is a more serious disease, tends to be localized in the lower respiratory tract. And then we have some viruses that will enter through the respiratory tract, but then they're spread systemically through the body. So German measles, mumps, hantavirus, we don't necessarily think of those as being respiratory viruses because the symptoms that we see in a sick individual are not associated with the respiratory system. But those viruses all enter through the respiratory tract and then spread. Then we have the alimentary tract. And again, the epithelial tissue is covered with microvilli, and we have mucus being produced. The mucus is important in our alimentary tract because it coats the food bolus that's moving through there and protects the walls of the alimentary tract. For a virus to enter through the alimentary tract, it has to be pretty tough. It has to survive exposure to antivirals in saliva. Then, when it gets down to the stomach, the pH is very low, about 2.5, and we produce the enzyme pepsin, which is very destructive. Then, when it gets to the small intestine, we have digestive enzymes from the pancreas and bile salts from the gallbladder. We also have IgA, and of course the mucus layer also helps to trap viruses. M cells are highly specialized cells present within the epithelium overlaying organized lymphoid follicles of the small and large intestine. They play a central role in the initiation of mucosal immune responses by transporting antigens and microorganisms to the underlying lymphoid tissue. In this way, the mucosal immune system encounters the limitless variety of antigens that enter the body through the gut mucosa and reacts by mounting specific mucosal and systemic immune responses. Now, the cytoplasm of M cells is very thin, and if you look at this EM, you can see that the villi are very short. They ingest and deliver antigens to that underlying lymphoid tissue. They also have a pocket that can harbor lymphocytes, and you can see that cell sitting right there in the middle is a lymphocyte, and actually in this case it's a macrophage, and they can provide entry for enteric viruses. So for instance, HIV can infect macrophages, and that's one reason why they think that HIV can enter very effectively through the alimentary tract, because there are many M cells in the lower colon, and this gives the virus a portal to susceptible lymphocytes. Okay, then we have other modes of entry too. Often viruses can enter through the urogenital tract as a result of sexual activity. We do have physical barriers though, mucus and low pH, for instance. But small tears or abrasions in the epithelium can provide a route of entry. Also, some viruses can produce local lesions in the epithelium, such as papillomaviruses, which cause genital warts. And some can gain access to underlying tissue and infect cells of the immune system, such as HIV or the neurons, which would be herpes simplex, as an example. Viruses can enter through the eye. The inner surfaces of the eyelids and the exposed part of the sclera are routes of entry for several viruses. Most often, infection is aided by abrasion. If it's a localized infection, it'll cause conjunctivitis or a pink eye. Systemic spread is rare, but does happen. So, for instance, herpes virus can infect the cornea, leading to blindness. And then this can be followed by spread through sensory neurons. But then we also have the skin. The skin is an effective barrier. Remember, we talked about that as part of the innate immune system. And the external surface is stratified squamous epithelial tissue, so the outermost layer is dead. The epidermis itself is devoid of blood and lymphatics, but the dermis has a good blood supply. So if there is a breach that's deep enough to get through the most external layer, the epidermis, and get down to the dermis, the virus can easily spread. So we see this happening a lot. Arthropod bites can provide a mode of entry, breaching the skin. Animal bites, for instance, rabies. The animal bites into muscle, the muscle has nerve endings, and the rabies makes its way to the nervous system. Punctures and other kinds of injuries all give the virus a mode of egress past the skin barrier and into the host itself. Okay, so next we want to talk about how the virus spreads. We can have localized infection, and with a localized infection, the virus produces injury predominantly near the site where it enters the host. So let's go back to our example of upper respiratory infections by rhinoviruses. Those viruses predominantly spread from one infected cell to the adjacent cells, and it remains pretty localized within the upper respiratory tract. We see similar things with skin infections by papillomaviruses. Again, it stays localized at the point of entry, so only adjacent cells are getting infected. So in these cases, the spread is a result of contiguous infection of adjacent cells. The virus rarely spreads beyond the epithelial layer. But then we have other modes of infection too. So we can talk about polarized release of virus. Okay, when we talk about polarized release or directional release of virions from polarized cells, remember epithelial cells are the cells covering all the surfaces, internal and external, of the body. Epithelial cells are what we call polarized. They sit on a basement membrane, so there's a basolateral surface. Opposite that is the apical surface. Now, the apical surface is the surface of an epithelial cell that will have things like the cilia and the microcilia. It's also where material such as mucus from goblet cells will be released, and so on. The functions of many kinds of epithelial cells are either secretory, meaning they release materials, usually from the apical surface, or they're absorptive. So consider in your alimentary tract where you're absorbing nutrients and so on. In that case, nutrients are absorbed through the apical surface, make their way through the cell, and then they're released through the basolateral surface so that the nutrients can get released to the body. So when we look at this, viruses can move depending on the polarization of the cell. So if a virus is released from the apical surface, meaning that the virus has entered the cell, it's replicated, now it's being released. If it's released from the apical surface, they're back where they started. Such a directional release can facilitate the spread of virus. So, for instance, enteric viruses in the feces. Think about poliovirus. It enters through the alimentary tract. It replicates, and one of the primary ways it's being spread is through fecal-oral contamination. So the virus is released back into the feces. Hepatitis A has a similar kind of pattern. Virus particles released from the basolateral surfaces of a polarized cell, that is the cell that's sitting on the basement membrane. Now the virus will be moving away from the defenses of the luminal surface, and it's moving away from the surface where the virus entered. Directional release is a major determinant of the infection pattern. In general, virions released at apical membranes establish a localized or limited infection. In this case, local lateral spread from cell to cell occurs in the infected epithelium, but virions rarely invade the underlying lymphatic and circulatory vessels. On the other hand, release of particles at the basal membrane provides access to the underlying tissues and may facilitate systemic spread. So if you look at these pictures, the EM at the bottom shows virus that infects from the apical surface and is also being released from the apical surface. You look at the bottom of the cell there, the basolateral surface, you don't see any virus being released. Now, if you look at the figure above, that cell has been infected with two different viruses, and one type of virus is being released from the apical surface, but the second virus, which you can see has those rod-like virions, is being released from the basolateral surface. Disseminated describes an infection that spreads beyond the primary site of infection. It often includes viremia and infection of major organs such as liver, lungs, and kidneys. Systemic describes an infection that results in spread to many organs of the body. So the thing to keep in mind here is that there isn't a clear-cut difference between a disseminated infection and a systemic infection. Systemic is just a term that's used for a disseminated infection that has really spread widely through the body. But they're not really mutually exclusive terms, so don't obsess too much over the difference between disseminated and systemic. When we're talking about disseminated infections, there are two major patterns of spread: hematogenous, or through the blood, and neural spread. Viruses that escape from local defenses to produce a disseminated infection often do so by entering the bloodstream. Virus particles may enter the blood directly through the capillaries, by replicating in endothelial cells, or through inoculation by a vector bite. Once in the blood, the virions have access to almost every tissue in the host. Hematogenous spread begins when newly replicated particles produced at the entry site are released into the extracellular fluids, which can be taken up by the local lymphatic vascular system. Lymphatic capillaries are considerably more permeable than circulatory system capillaries, and that facilitates virus entry. The lymphatic vessels join with the venous system, giving virus particles in the lymph free access to the bloodstream. In the lymphatic system, virus passes through the lymph nodes, where they encounter migratory cells of the immune system. Virus pathogenesis resulting from the direct infection of immune system cells, such as what happens with HIV, is initiated in this fashion. Some viruses replicate in the lymphoid cells, and progeny are released into the blood plasma. Concentration of virus in blood is determined by rates of synthesis in permissive tissues and by how quickly they are released into and removed from the blood. Most viremias are of short duration. However, some viral infections are noteworthy for long-lasting presence of infectious virus in the blood. Hepatitis B and hepatitis C, for instance, can cause viremias in hosts that last for years. Viremias are of diagnostic value and can be used to monitor the course of infection. However, infected blood presents problems for health care in terms of transfusions and blood clotting factors and so on. And we can talk about primary viremia and secondary viremia, but let's look at this in the following picture. So here you see replication at the site of entry. And again, this can be the skin, the mucous membrane, the respiratory tract, and you get infection. So with some viruses, the virus remains localized. Remember, we talked about that. So herpes simplex virus, papillomaviruses, they tend to be shed from the surface where they entered. But we can have entry of viruses after replication at the site of infection into the blood. And this is our primary viremia. So the virus can be spread now to many different tissues in the body. So we're having a systemic dissemination. And depending on the virus, various replication sites may be the targets. Could be muscle cell, could be liver, spleen, or blood vessels. But the virus will go through an additional round of replication at these sites. But eventually the virus is shed from these sites, and we have the secondary viremia. At this point, the virus may be shed in the blood, and we see hepatitis B virus and so on, HIV virus. But many times the virus will move on to even more tissues after the secondary viremia. And that's when we get into issues of skin, mucous membrane, lung, kidney, gastrointestinal tract, and brain. And it's at these replication sites after the secondary viremia where we start to see transmission to other hosts. So, for instance, if we go to the lungs, hantavirus is released that way. If we go to the gastrointestinal tract, we have various enteroviruses and reoviruses and so on that are released that way. So we have very different patterns of pathogenesis, even though the basic steps are the same in terms of entry, replication, primary viremia, additional replication, secondary viremia, more replication, and then shedding or transmission to other hosts. So these different viruses address these issues in different ways, and it results in different patterns of pathogenesis. Let's look at another example. So let's look at a specific example. This is pathogenesis of mousepox virus. In a laboratory situation, they would inoculate the virus into the foot pad. And that's where you get your initial replication. Eventually, the virus is released into the bloodstream. That's your primary viremia. The next stop is going to be the spleen and liver. That's where you get that next round of replication. But then the virus is released back into the blood, and you have your secondary viremia. Now, in this case, the virus returns to the skin, but it's moved way beyond just the foot pad. And you'll get lesions on the skin all over the body. Neurons are polarized cells with functionally and structurally distinct processes, the axons and dendrites, that can be separated by enormous distances. In an adult human, the axon terminals of motor neurons that control stomach muscles can be as much as 50 centimeters away from the cell bodies and dendrites in the brainstem. Viral replication usually occurs first in non-neuronal cells, with virions subsequently spreading into sensory or motor nerve fibers that innervate the infected tissue. Once in the peripheral nervous system, the virus can spread among neurons connected by synapses. Infection can then spread to ganglia and eventually to the brain and spinal cord. An important component of neuronal infection is the movement and release of infectious particles in polarized neurons. Directional release of infectious virus from the neurons affects the outcome of infection. So let's look at this in the next slide. So when we're talking about infection of the nervous system, there are three important terms. Neurotropic virus can infect neural cells. Neuroinvasive virus can enter the central nervous system after infection at a peripheral site. And a neurovirulent virus can cause disease of nervous tissue manifested by neurological symptoms and often death. And this gets tied into the aspect of directional release of infectious viruses. Remember, I said that nerve cells are polarized, and we see directional release of virus particles, and that can affect the outcome of infection. So let's look at some examples. First, herpes simplex, the cold sore virus. It has a low neuroinvasiveness of the central nervous system, but a high neurovirulence. It always enters the peripheral nervous system, but rarely enters the central nervous system. So what I'm talking about here is the first time you get infected with herpes simplex, it replicates in the tissue, the peripheral tissue, and then the sensory nerves that innervate that tissue become infected. The virus can remain latent in those nerve cells. Then under certain conditions, say stress, something like that, the virus gets reactivated and it makes its way back to that peripheral site where it first entered, and you get a cold sore. That's why you get a cold sore forming in the same place every time, because it's related to this polarization of the nerve cells. So, developing a cold sore is an annoyance. But fortunately, with herpes simplex, it has low neuroinvasiveness. Only on rare occasions does herpes simplex virus enter the nervous system. When it does, the consequences are always severe and very frequently fatal. Now, mumps has a high neuroinvasiveness but a low neurovirulence. Most infections lead to invasion of the central nervous system, but the neurological disease is mild. I can speak from personal experience here. As a kid, I earned my immunity to all the childhood diseases the hard way by getting them and developing immunological memory. So I had the mumps. And of all the childhood diseases, so I had both types of measles, I had chicken pox and mumps. Mumps was the one that stands out most because I can distinctly remember, even though I was just a little kid, that I had a terrible headache when I had the mumps. And that's because of the neuroinvasiveness. Fortunately, though, the neurological disease is very mild, and children usually recover. You can see I'm still around, so obviously I did. Okay, then we can talk about rabies virus. It has a high neuroinvasiveness and a high neurovirulence. It readily infects the peripheral nervous system, and it usually spreads to the central nervous system. And when it does that, it causes nearly 100% lethality unless there's an antiviral therapy that's administered in time. So in this case, remember, the virus is usually introduced by an animal bite. That bite introduces the virus past the skin barrier down into muscle tissue where it first replicates, but then it's able to innervate the nerves in the muscle tissue, makes its way to the central nervous system, and eventually will cause fatal encephalitis. Okay, so we need to talk about levels of tropism as well. We can talk about viral tropism, or the predilection of the virus to infect certain cell types or tissue types. At the level of the cell, at the level of the tissue, or at the level of the host. Most viruses do not infect all the cells of a host, but are restricted to specific cell types of certain organs. Viral tropism can be divided into 3 distinct categories, depending on the physiological level at which it is measured. Tropism in which the virus replicates in one cell type but not another is known as a cellular tropism. Tropism in which the virus replicates in a particular tissue or organ but not another is known as tissue tropism, and tropism in which the virus replicates in one host species but not another is known as host tropism. And you can see some of the examples here. So HIV normally infects macrophages but not neurons. Influenza normally infects lung tissue but not brain tissue. And myxoma virus normally infects rabbits but not humans. So those are some examples of these different levels of tropism that we can see. Okay, when we're talking about Viral tropisms. There's some parameters we need to discuss. So first, there's susceptibility. That's whether or not an animal can become infected. This is usually determined by the distribution of receptors for entry. For instance, there are thousands of species of mosquitoes out there, but when you talk about a particular species, a particular type of virus, only certain species will be vectors, and a lot of that has to do with susceptibility determined by receptors in the midgut of the mosquito. Some mosquitoes do not have the appropriate receptors to become infected. Or, for instance, why don't mosquitoes transmit HIV? I get asked that a lot. Mosquitoes don't have the appropriate receptors. They're not susceptible to HIV, which I'd say is a damn good thing. Then we have permissivity. And this requires the virus to actually be able to replicate and spread in the animal, so beyond just becoming infected. And you usually need intracellular gene products to complete the infection. So you might need specific cellular proteins, such as an enhancer or a protease, something like that. So as an example, the flu is usually limited to the upper and lower respiratory tract because it requires a protease to cleave the hemagglutinin precursor. Expression of this protease is limited to the respiratory system. However, a mutant virus that can use a more ubiquitous intracellular protease could have altered tropism and virulence. Then you have to have accessibility. Does the potential host animal and the virus ever come into contact? Are they will the virus be able to access that host? And then, of course, we have local immune responses, the innate immune system, and so on, that can also play a role in determining tropism. So virus shedding is the release of infectious viruses from an infected host, and of course that's required for transmission to new hosts. Localized infections will usually result in shedding taking place from the primary site of replication. Release of virions that cause disseminated infections can occur at many sites. So think back to that slide that we looked at earlier when we looked at primary and secondary viremias and shedding and so on. Now, effective transmission from one host to another depends on concentration of particles released and the mechanisms by which the virions are introduced to the next host. The extent of virion survival in the environment also influences the efficiency of transmission. So consider respiratory shedding. Aerosols produced from speaking, breathing, coughing, sneezing all can transmit virus. For instance, with a sneeze, you can be releasing 20,000 droplets. A cough, several hundred. And the droplets can travel a distance inversely related to size. Nasal secretions contaminate hands and tissues, and another person can pick it up this way. So, we're seeing this with how we're trying to avoid coronavirus. You wear a mask primarily to protect others from all the virus that you may be spreading from your respiratory system. Saliva is another way that viruses shed. Viruses that replicate in lungs, nasal mucosa, and salivary glands, such as the Epstein-Barr virus, which causes mono, are shed in saliva. Rabies and feline leukemia virus are also shed in saliva. Viruses can be shed in the feces. Viruses that are spread this way have to be very resistant to environmental conditions. They must survive in a dilution of water and drying. Poor sewage treatment, contaminated irrigation systems, and situations like that contribute to the transmission of virus that's shed in the feces. But examples of important viruses like this would include poliovirus and hepatitis A virus. Remember, hepatitis A virus is the type of hepatitis virus that results in restaurants getting closed down. And what can happen is a infected person working at a restaurant may not wash their hands properly, and they transfer virus to the food of the patrons, the restaurant patrons. They ingest the virus, and it infects them through their alimentary tract. Virus can be shed in blood. Viremias, remember, are common. Arthropods, transfusions, childbirth can all result in transmission of virus through blood. We get some diseases such as Ebola that cause fatal hemorrhagic fevers, and these individuals release a lot of blood and body fluids. That's why with diseases such as Ebola, healthcare workers are often amongst the first to die because they're the ones who come into contact with these infected bodily fluids, sometimes before it's known that there's a problem with Ebola in an area. Milk can be a vessel for virus transmission. West Nile virus is spread through mother's milk. Mouse mammary tumor virus is transmitted this way. Hantaviruses and arenaviruses are spread through the urine. What happens here is that rodents become persistently infected. They shed the virus in their urine, and then the virus is very resistant to environmental conditions. So it survives drying, and then it can be aerosolized when the dried urine and also the dried feces get aerosolized. People can breathe it in. HIV and hepatitis B are spread through semen. And skin lesions, herpes simplex, pox viruses can be spread by direct contact through skin lesions. We also need to consider viral virulence. Virulence is the capacity of the infection to cause disease. It's a quantitative statement of degree or extent of pathogenesis. And there are three different terms that we often see being used. If we refer to a virus as virulent, we mean that it causes significant disease. If it's avirulent, it causes no disease. And if it's attenuated, it causes reduced disease. So, for instance, many vaccine virus strains are attenuated. We can measure these things in a number of ways. We can look at endpoint methods such as LD50. We can look at mean time to death or appearance of symptoms. We can do histological evaluations of tissue damage. With HIV, we look at reduction in the blood concentration of CD4 lymphocytes. Or we can assay liver enzymes that are released into the blood as a result of virus-induced liver damage. So that's a common thing we see happening if a patient or if you take your pet to the vet, for instance, or you go to your doctor, and it appears that you have some sort of infection going on. One of the things that the doctor will do is take a blood sample. And if they see certain liver enzymes are elevated, that's a sign of a potential virus infection. They don't know what virus is causing it yet. It's just a sign that that kind of infection is going on. Virulence depends on dose and route of infection, and other kinds of factors play a role as well. And genetic determinants of the virus are important. So, for instance, in the illustration on this slide, we're looking at two types of avian influenza. LPAI is low pathogenic avian influenza, and HPAI is high pathogenic influenza. You can tell because with the high pathogenic influenza, our little rooster friend is laying on his side. Okay, so with the avian influenza, you have the precursor hemagglutinin that has to be cleaved into HA1 and HA2. This cleavage is dependent on certain kinds of enzymes. In the low pathogenic viruses, there's it's a monobasic cleavage site that is cleaved by trypsin-like proteases only, and that limits the replication of these viruses to sites in the body where these enzymes are expressed, specifically the respiratory and intestinal tracts. In contrast, the precursor, the HAO of highly pathogenic avian influenza viruses, can be cleaved by ubiquitously expressed furin-like proteases, which facilitate systemic replication in chickens. So a small genetic difference between the different strains of virus can determine virulence. Okay, when we're talking about pathogenesis, we're talking about a single individual, the replication of the virus within that host. Epidemiology, on the other hand, is the study of the occurrence of a disease in a population. Remember, viruses only survive if they spread from one susceptible host to another. And epidemiology takes in all kinds of factors. There's the mechanism of transmission. What are the risk factors for infection? What's the size of population needed for transmission? Geography plays a role, seasonality plays a role, and means of control all affect the epidemiology of a disease. So let's look at some of these factors. First, we have forms of transmission. Zoonoses are diseases shared by animals and humans and/or insects, as opposed to an anthropogenic virus that only infects humans. If animals are involved in the replication cycle, that really changes things in terms of epidemiology. We can talk about vertical transmission. That's the transfer of viruses between parent and offspring. This can happen during gestation, where the virus crosses the placenta. It can occur during birth. So sometimes, for instance, with HIV, transmission of the virus from the infected mother to the child happens more commonly at birth, when the child is exposed to infected blood, rather than during gestation. Fomites are contaminated objects, and usually a virus that can withstand environmental conditions would be transmitted that way. Iatrogenic transmission is when the activity of a healthcare worker leads to infection, and nosocomial transmission is when an individual is infected while in a hospital or healthcare facility. So let's look at these different factors a little bit more closely. So again, with the zoonosis, this is when we're not just talking about a virus being transmitted from person to person, but we have various types of animals that can get involved in this. And there are a lot of different important viruses that involve animals and/or insects in their transmission cycle. As I said, this makes the epidemiology of a virus much more complex than if we're just talking about person-to-person transmission. Okay, then we have vertical transmission. So again, that's from the parent to offspring. And, we saw the, truly tragic results of Zika virus. Zika virus causes different types of brain damage in babies, not just microcephaly, according to some new reports. Brazilian researchers found as many as 1 in 5 babies born with brain damage caused by Zika had normal-sized heads. That means that babies who may seem normal may in fact suffer from serious conditions that doctors and parents may not notice until they get older. The findings show that if a pregnant woman is infected with Zika, even if the baby is born with a normal-sized head, the child could have severe brain damage. But, it's just one of the more recent notable examples of vertical transmission of virus. Fomites are any surface or object that can become contaminated with viral or bacterial pathogens and that can act as a vehicle of transmission. Studies found that most viruses survived longer on non-porous surfaces, such as metal or plastic, and that respiratory viruses, such as influenza virus, rhinoviruses, and coronaviruses, could remain viable for hours or even days on these surfaces. Under the right conditions, enteric viruses like norovirus and hepatitis A have been known to survive on fomites from weeks to months. I'm here to tell you, it's a germy world out there, and most people touch their faces 10 to 20 times each hour, providing ample opportunities for pathogens to move from fomite to fingers to face. So, kids, take it from Dr. Paulson. Wash your hands. Iatrogenic transmission occurs when the activity of a healthcare worker leads to infection of the patient. Improper use of syringes, needles, and medication vials during routine healthcare procedures, such as administering injections, have resulted in more than 50 outbreaks in U.S. hospital and non-hospital settings, with transmission of blood-borne viruses, including hepatitis B and hepatitis C, to more than 600 patients. These outbreaks resulted from failure to follow basic infection control procedures and aseptic technique in injection safety, including reusing syringes, contaminating multi-dose vials with unclean syringes, using single-dose vials for multiple patients, reusing end caps from single-use syringes, using fingerstick devices on multiple patients without cleaning, and using blood sugar measuring devices on multiple patients without cleaning. Nosocomial transmission occurs when an individual is infected while in a hospital or healthcare facility, but it's not necessarily from the direct activities of the healthcare worker. It's estimated that healthcare workers, patients, and visitors are responsible for spreading about 80% of common infections contracted in healthcare settings. Further research has shown that items of clothing can play a role. For instance, neckties worn by doctors and other medical personnel are likely carriers of infection-causing bacteria. There was a study done at a Queens hospital in 2004 that found that 47% of the ties worn by medical staff at the hospital harbored illness-causing bacteria. The study also noted that ties worn by doctors and other clinical staff were 8 times more infectious than security guards' ties. So, just requiring changes in the kind of professional clothing that healthcare workers are utilizing can make a big difference. The movement to scrubs has been really helpful. Hospitals are now required to provide an adequate supply of scrubs to medical staff to ensure that they change their clothes frequently. And also, they ban the wearing of uniforms outside of the hospital or other healthcare setting to prevent transmission of disease. In a study of more than 2 million people in the UK and USA between March 24th and April 23rd of this year, it was noted that frontline healthcare workers had at least a threefold increased risk of reporting a positive COVID-19 test and predicted COVID-19 infection compared with the general community, even after accounting for other risk factors. Post hoc analyses showed that Black, Asian, and minority ethnic healthcare workers are at especially high risk of infection, with at least a five-fold increased risk of COVID-19 compared with the non-Hispanic white general community. Among frontline healthcare workers, reuse of PPE or inadequate PPE were each associated with subsequent increased risk of COVID-19. Although healthcare workers caring for patients with COVID-19 who reported inadequate PPE had the highest risk of infection, increased susceptibility to infection was evident even amongst those reporting adequate PPE. Frontline healthcare workers who worked in inpatient settings, where providers most frequently reported PPE reuse, and nursing homes, where providers most frequently reported inadequate PPE, had the greatest risk. Non-white healthcare workers were disproportionately affected by scant PPE adequacy and more likely to work in clinical settings with greater exposure to patients with COVID-19. With cases of COVID-19 climbing, nursing homes are hotspots because they combine numerous risk factors for transmission: congregate living, a mostly elderly population with underlying health conditions, and inadequate staffing, PPE, and infection control for emergencies. At least 75,000 Americans in nursing homes and other long care facilities have already died from COVID-19, and the devastation is far from over. After a decrease earlier this summer, the death toll is now rising once again, and as the country heads into the fall and then flu season, millions of Americans who require institutional long-term care remain at the greatest risk. There are other mechanisms of transmission as well. Aerosol, food and water, bodily secretions, birth. One thing that you may not think about as much would be transplants. In 2004, we had a case where a patient in Houston with undiagnosed rabies was a donor to 3 recipients. All 3 tissue recipients later died. A 4th later died from an arterial graft. In 2005 in Germany, we had another case of rabies passed through transplantation where 3 recipients died. Okay, then there are factors that promote transmission. Of course, the stability of the virus and aerosols and secretions. Asymptomatic shedding is really important. What that means is that a person who isn't exhibiting symptoms of the disease may be shedding a large amount of virus. We're very concerned about that, for instance, with coronavirus, that particularly very young people may be asymptomatic, but they could be shedding virus and transmitting the disease to other people. So we don't always have to have symptoms in order to have viral shedding. And an ineffective immune response plays a role too. What we see in our modern society is that there's a larger and larger segment of the population that have reduced or ineffective immune responses. Many times this is intentional. So, for instance, if you receive a transplant, let's say you get a kidney transplant, for the rest of your life you are going to be receiving drugs that suppress your immune system so that you won't reject that organ. And then there are diseases that cause immune suppression, for instance, HIV. So we see a larger and larger component of the population who have a reduced, ineffective, or defective immune response, and that can promote transmission of disease. Geography and season play a role in transmission of disease. Of course, if the virus is transmitted by an insect vector, the virus will only be transmitted during the portions of the season when that insect is active. So, for instance, in the temperate region, we only see viruses transmitted by mosquitoes being transmitted during the late spring to very early fall, and then the mosquitoes die off and we don't have any more transmission till the following spring. We see that respiratory infections predominate in winter, and enteric infections tend to be more abundant in the summertime. It's just a common thing. The school year plays a role as well in transmission. People go out during the summer, travel around, they may pick up new pathogens. They come back, they send their kids to school, and now you've got all this mixing that's going on. Home heating can contribute to the transmission of certain kinds of viruses as well. There are risk factors that have to be considered in addition to some of these other factors we've been talking about. Age can play a role. With certain viruses, the very young or the very old may be at greatest risk. It depends from disease to disease. The overall health of the individual can play a role. Their immunity, their occupation. Certain occupations can put you at risk. Travel, lifestyle, children. Do you send your kids to schools or daycare centers where they may pick up a disease and bring it home? Of course, sexual activity can be a risk factor for certain kinds of viruses. And we can see this in this graph at the bottom. These are two different types of influenza virus. And with the one on the left, you can see that males versus females tended to be infected more often. And then if we move over to the right side, we can see that it remains much more equal in the distribution. And this differential exposure that we see between males and females may have to do with gender-associated practices and norms, meaning, what kind of work are the males doing versus the females? Is that bringing them into contact with the virus? There could be biological differences between males and females in the clinical course, post-exposure and infection. And differential healthcare seeking and access behavior between males and females, leading to surveillance detection bias. So, there are a lot of different things that can act as risk factors, and it will vary from virus to virus. The critical community size, or critical population size, is the minimum size of a closed population within which a human-to-human, non-zoonotic pathogen can persist indefinitely. That means a disease that is not spread in part by animals, because animals will change all of your calculations, all of your modeling systems, and so on. So we're talking just about viruses that are transmitted from person to person. When the size of the closed population falls below the critical community size level, the low density of infected hosts causes extinction of the pathogen. This epidemiologic phenomenon was first identified during the measles outbreaks in the 1950s. The critical community size depends on the speed of transmission, how long until a person who has recovered remains immune, the fatality rate, and the birth and death rate in the general population. So these factors all play a role in how large or how small the critical population size will be. And then we also have herd immunity. This is when the prevalence of immunity to a virus in a population reaches herd levels. That is, enough people are resistant to keep an epidemic from growing. It's really better to consider herd immunity when you're talking about vaccines. But herd immunity as a strategy has been considered by some countries as a way to combat the current COVID-19 crisis by ignoring risky behavior in the hope that infected people will survive, become resistant, and reduce the susceptible population. This is an approach that will increase the deaths and disability caused by the virus, and it really should be avoided. So, for instance, when we're talking about COVID-19, this is a highly infectious virus, so the minimum level to reach herd immunity, where we'd expect newly infected people to pass the virus to less than one additional person, or the R0 is less than 1, is thought to be about 60% of the population. It's estimated that right about now, the U.S. has only reached about 3% of our population having been infected, and we're already up over 190,000 deaths. Let's consider Sweden. This is a major country that tried herd immunity earlier this year as its national strategy. As part of the plan, Swedish officials allowed restaurants, preschools, and grade schools, public transportation, public parks, hair salons, yoga studios, gyms, malls, movie theaters, and ski slopes to remain open without restrictions. However, they did limit public gatherings to 50 or fewer people, and they closed their high schools and universities, closed museums, canceled public sporting events, and banned visits to nursing homes. Social distancing, however, was voluntary. The results of Sweden's decision to attempt herd immunity were disappointing and devastating. By the end of May 2020, the nation recorded nearly 41,000 COVID-19 cases, resulting in more than 4,500 deaths in a population of 10 million, compared with Scandinavian neighbors Norway and Finland that counted fewer than 600 COVID-19 deaths combined. Sweden's state epidemiologist and the architect of this herd immunity plan admitted at a press conference in early June that too many people died too soon as a consequence of this strategy. The United States has a large vulnerable population, differing strategies at community, state, and national levels for dealing with COVID-19, and a healthcare system with shortcomings and inequities. So it will be very difficult to reach the necessary level of immunity without an effective vaccine. A bigger question still needing an answer is whether or not COVID-19 infection yields long-term immunity. For herd immunity to work, prior exposure to the virus has to prime the immune system to produce a strong response to all future contact with the virus, and in turn make the person less infectious. We don't know if that happens with this specific coronavirus. So the herd immunity strategy is a shaky foundation on which to base a disease-fighting strategy that could fail and lead to sort of a whack-a-mole series of outbreaks for a long time. Okay, then we have means of control. Now, obviously, if your disease is transmitted by a vector, then you can go about vector elimination. So in this picture, this individual is spraying insecticide using a fogging machine during a vector control campaign in Thailand. And in this case, they were trying to kill off vectors of malaria. But we have other methods as well. We'll talk about quarantine and isolation in this lecture. Immunization and antivirals will be considered in the next module. Okay, I should point out as we get started that with the COVID-19 situation, you'll find that the terms quarantine and isolation have been used almost interchangeably, but they actually do have different definitions. Isolation is when you're isolating a person who is sick, who has a communicable disease, from those who are healthy. Isolation restricts the movement of the sick person to help stop the spread of the disease. For instance, we use isolation in hospitals for patients with infectious tuberculosis. Quarantine is used to separate and restrict the movement of people who may have been exposed to a disease to see if they become ill. These people may have been exposed to a disease and do not know it, or they may have the disease but do not show symptoms. Quarantine can also help limit the spread of communicable disease. The federal government derives its authority for isolation and quarantine from the Commerce Clause of the U.S. Constitution. So, under this act, the U.S. government is authorized to take measures to prevent the entry and spread of communicable diseases from foreign countries into the United States and between states. The authority for carrying out these functions on a daily basis has been delegated to the Centers for Disease Control and Prevention, or the CDC. Under the procedures required by the Public Health Service Act, the list of diseases for which quarantine is authorized must first be specified in an executive order of the President on recommendation of the HHS Secretary. Since 1983, this list has included cholera, diphtheria, infectious tuberculosis, plague, smallpox, yellow fever, and viral hemorrhagic fevers. It was amended in April 2003 to include SARS. In the midst of the 2003 global SARS outbreak, HHS amended the quarantine regulations to incorporate the executive order that defines the list of diseases for which quarantine is authorized. This action streamlined the process for adding new quarantinable diseases in the future by eliminating the need to publish the list in both an executive order and regulations. Future revisions to the list of quarantinable diseases will require only an executive order, and you can see that MERS and COVID-19 have been added to the list. This photograph from about 1930 shows a person sitting in New York's Ellis Island. At that time, anyone arriving at the immigration station on Ellis Island who appeared to have a communicable disease was immediately segregated. If diagnosis confirmed the suspicion, that person was quarantined in a hospital until he or she was no longer contagious. You saw this if you ever watched Godfather 2, when young Vito arrived in New York. They determined that he had TB, and he was kept in isolation until he had stopped showing symptoms of the disease. U.S. quarantine stations are part of a comprehensive system that serves to limit the introduction and spread of contagious diseases in the U.S. We have stations located at 20 ports of entry and land border crossings where international travelers arrive. You can see this on the map. They're staffed with medical and public health officers from the CDC and managed by the CDC's Division of Global Migration and Quarantine. These health officers decide whether ill persons can enter the United States and what measures should be taken to prevent the spread of contagious diseases. CDC has the legal authority to detain any person who may have an infectious disease that is specified by the executive order to be quarantinable. If necessary, CDC can deny ill persons with these diseases entry to the United States. CDC can also have them admitted to a hospital or confined to a home for a certain amount of time to prevent the spread of disease. In a desperate race to stunt the spread of coronavirus, millions of Americans were asked to do what would have been unthinkable only a few months ago. Don't go to work, don't go to school, don't leave the house at all unless you have to. The directives to keep people at home, which began in California in mid-March, quickly swept the nation. Altogether, more than 300 million people in at least 42 states, 3 counties, 10 cities, the District of Columbia, and Puerto Rico were being urged to stay home. In Virginia, Governor Ralph Northam issued a statewide stay-at-home order in mid-March. We are now in phase 3 of reopening the state as businesses slowly reopen. But, the executive order for phase 3 includes the caveat, we must remain cautious, continue teleworking whenever possible, wash our hands frequently, do not touch our faces, and wear face coverings and masks. So, shelter in place is something that was added in our response, in addition to quarantine and isolation. And of course, social distancing. That's become the new normal for us, where you stay 6 feet apart and wear a face covering to reduce the possibility of transmitting the virus. And social distancing, along with wearing the face mask, has been shown to be quite effective at reducing the transmission of the virus. So as we complete our discussion of isolation and quarantine and all those fun things, I just want to stress the point that this is a difficult time for everybody. Fear and anxiety about a new disease and what can happen can be overwhelming and cause strong emotions in both adults and children. Public health actions such as social distancing can make people feel isolated and lonely and can increase stress and anxiety. However, these actions have been shown to be able to reduce the spread of COVID-19. But, I just want to emphasize to everybody, you want to try to cope with stress in a healthy way that will make you and the people you care about stronger. And if you're feeling that you're struggling, you're having any problems, don't hesitate to reach out and get some help. There's a lot of help out there for you, including right here at Virginia Tech. The rigid cell walls of plants can make it difficult for a virus to enter, but there are many routes by which plant viruses can be transmitted. Seeds may transmit virus infection either by external contamination of the seed with virus particles or by infection of the living tissues of the embryo. Transmission by this route leads to early season outbreaks of disease in new crops. The disease may be focal in distribution initially, but may eventually be transmitted to the remainder of the crop by other mechanisms. Vegetative propagation and grafting techniques are cheap and easy methods of plant propagation, but provide the ideal opportunity for viruses to spread to new plants. Also, many different types of organisms, such as aphids and nematodes, can act as vectors. Transfer by insect vectors is a particularly efficient means of virus transmission to plants. Mechanical transmission refers to virus infection through wound openings that are produced by animal feeding or farming machinery. Even rainstorm and hailstorms have led to the transmission of virus when the plants were closely spaced. In the laboratory, when you want to induce an infection in a plant, we very commonly will brush the plant surface with an abrasive to cause a point of entry. In order to infect the entire host plant, viruses have to move from the initially infected cells to neighboring cells and then spread from inoculated leaves to other leaves through the plant vascular system. These two processes involve different mechanisms. Although plant cells are surrounded by thick layers of cell wall, adjacent cells are linked by cytoplasmic connections called plasmodesmata. Recent experiments have shown that plant viruses encode a special protein called movement protein that increases the pore size of the plasmodesmata, thus allowing the virus to move across the gap more readily. Long-distance translocation occurs through the movement of virus through the phloem of the vascular system. Systemic symptoms of a viral infection often occur in new growth areas, such as young leaves at a plant tip or young roots. In many viruses, long-distance translocation requires a functional capsid of the virus, probably to protect the viral nucleic acids from the adverse environment in the phloem tissues. Not all viral infections cause visible symptoms on infected plants. Also, it is possible for an infected plant to recover from a virus infection. The initially infected leaves show obvious symptoms, but the newly emerged leaves after the recovery will have no symptoms, even though the virus can replicate to a high level. Local lesions can develop near the site of virus entry on leaves or fruit. The most common symptoms are chlorotic or necrotic spots and ring spots, ranging from single to concentric rings. Local symptoms are usually the result of interaction between host defense responses and virus infections. These defense responses include rapid death of injured cells from a hypersensitive reaction and physiological changes that limit virus dissemination. The constant battle between a virus and a plant host is illustrated by the symptom of concentric rings. Each ring is indicative of an ongoing battle. Systemic symptoms result from the systemic invasion of a plant host by a virus. When viruses reach the phloem, viruses can be transported throughout the plant, causing this systemic infection. Mosaic patterns are common with systemic infections. Mosaic simply means an alternating pattern of tissues with two different colors. The colors can be light green, dark green, yellow, golden, or even white. If the borders between areas are sharp, then the symptom is referred to as a mosaic. If the borders are diffused, it's called mottle. On monocot plants, the symptoms are more likely to be of stripes or streaks due to the parallel structure of the veins. Yellowing can result when infection of the phloem disrupts the transport of nutrients and water throughout the vascular system. Stunting and dwarfing of the infected plants are another common symptom of systemic virus infection. Interference with plant growth hormone levels is probably the most important factor causing dwarfing. Other factors include reduced photosynthesis and reduced translocation of nutrients. Change in plant growth hormone is also responsible for malformation of plant parts. Symptoms may include stunting for the whole plant, leaf malformation, twisting, curling, shoestring-like leaves, galls or tumors, and leaf roll. Occasionally, the entire infected plant may become necrotic. Symptoms usually start along the veins and whole leaves, or the plant itself may die eventually. This is usually caused by a delayed hypersensitivity reaction. The infecting virus has moved out of the entry site and systemically invaded the entire leaf or plant before the hypersensitivity reaction kicks in.